Hello everyone, you've joined me trying to look casual in my flat kitchen. Since we last spoke, I've undergone the trim. It was a sterling job by my girlfriend, however I now look like this, so that's fun. I'd like to thank everyone for the excellent response to the Skittles practical. It was really nice actually seeing some of you send in what you've done, and it was really pleasing to see that you didn't just watch it just to laugh at me, so there we go. I've got another experiment for you. This one's going to take a little bit longer, but let's get on to it. 4-year-old Amazon lab coat's back, this time with a clap. So with this experiment, I thought I'd continue the theme of diffusion from the Skittles one. We're going to do osmosis, but this time with eggs. Now, because of the eggs, it's going to take a little bit longer. So I've got a quick budget option for you with potatoes instead, which also actually has the benefit of being a required practical at GCSE. The egg one is cooler though, so I'm going to persevere with the egg one and mention towards the end how you can do it with potatoes instead. This experiment is going to be 3 stars. Still haven't worked out what the stars exactly mean, but I'm going to rate it 3 stars because A, we're going to have some acid, some vinegar, and B, it's going to take a little bit longer. We're actually going to have the eggs in vinegar for a couple of days before we even get going with the experiment. So it's a little bit more commitment, and therefore 3 stars. You will need eggs. You'll need 2 eggs, so we can compare the 2 different solutions. Now, the only place that I could find eggs was Waitrose, so I'm clearly doing well, clearly being fancy. But what part of the egg do we actually need? It's not the shell. There's actually a thin membrane part that is kind of inside the shell. You can see it here if I break it apart. It's a kind of a thin membrane. We want the egg and that membrane. We don't want the shell. So we're going to need to get rid of the shell of 2 eggs. You will need some way of holding 2 eggs in 2 different liquids. I've used wine glasses because I'm fancy. You will also need water. You will need something very sugary, like golden syrup. Or you can use sugar and dissolve it in water, but golden syrup's way easier and way more sugary and tasty. You'll need some distilled malt vinegar, so it's clear and it doesn't colour the eggs, and you can also see what's happening, which is quite cool. You'll need a clear container which can hold 2 eggs and vinegar, like this Pyrex jug. You'll need some form of scale or balance. An electronic one is better, but if you've got an analogue one with a tray and a needle, go with that. Okay, on to the experiment. So the first step, as I've said, is to take that shell off the egg. And to do that, we're going to use the vinegar. So I'm going to take my container, I'm going to take my malt vinegar, and I'm going to take 2 eggs. I'm going to put the eggs into the container. I'm going to take the malt vinegar, okay, and pour it on top. It would also help if I got vinegar without that little hole at the top, so it'd be a little bit faster. It will obviously smell of vinegar, and it will obviously smell of egg, so enjoy that. It takes about 2 days for the shells to dissolve, so we're going to leave them in here for 2 days, and then on the 3rd day we're going to start our osmosis experiment.
2 days later.
Okay, so it's now been 2 days since I put the eggs in the vinegar. I did actually change the vinegar yesterday. I panicked a little bit. I actually would recommend don't change the vinegar, but obviously I have changed the vinegar. Here's what we're ending up with. I put some cling film over the top because I was a little bit put off by the smell, and I like vinegar. And these are really, really weird. They are shellless eggs. I'm going to clean them up and I'll check back in. Okay, so I've cleaned these eggs up now, and they're really bizarre. I would recommend, even if you don't do the next part of the experiment,
just make yourself a shellless egg. It's very, very weird just feeling the membrane. Bizarre. It's like a bouncy ball, but delicate. If you can put up with the weird gunk and the smell of vinegar. So, we're going to crack on now with the actual main part of the experiment. Okay, now we've got our 2 shellless eggs. We're going to be placing them in golden syrup and water to see the effect that it has on the egg. Now, to do that, given the fact that we're going to leave them there for a day, we're probably going to forget those numbers. So we're going to need a good old results table. Now here's one that I've quickly made up. It's bad because I haven't used a ruler. However, you can see here I've got a row for the water and a row for the golden syrup. And I've got a column for the mass before and the mass afterwards, because we're going to be interested in the change. So to measure the mass, I'm going to take out my balance. Okay, set it up. Okay, now I want to make sure that it's in grams. Here it is. And I'm going to put my shellless egg on there. So this first one weighs 78. And this egg is going to go into the water. This egg, okay, is going to go in the golden syrup. Before it weighs 76. So they're actually 2 quite similar masses of eggs. So now I've weighed my eggs. I'm going to take my 2 containers, which, because I'm well posh, are wine glasses. I'm going to place an egg in each. This one was the water egg. Okay. Oh, that's nervous wracking. This one was the golden syrup egg. The only reason I'm using wine glasses, by the way, is because my glasses are quite narrow, and I was worrying that I wouldn't be able to get them out. But thinking about this, these are quite narrow themselves, so we'll see how this goes. Golden syrup is a bit of a pain to get into. Okay, but what I'm going to do is pour the golden syrup in over the egg. Okay. Now this looks like I've really lost the plot. You don't see this type of thing on Gordon Ramsay, do you? Okay, I'm going to right pig's ear of that, but there we go. So that is an egg in a wine glass of golden syrup. Golden syrup, still very tasty. Water. I'll fill this up with water. Now, ideally, you would have the same volume of water to make sure that that's a control variable as the police goes past. This is so that volume is a control variable, and it means that it's not going to affect the experiment. And that's quite tricky. I can get down in line and see that they're roughly equal. I'm now going to leave these for a day, and we're going to, or at least overnight, and I'm going to come back and I'm going to see how it goes. Now, part of the other reason why I use wine glasses is so I can occasionally peek in and see if there's any effect. But let's check back in with that tomorrow. Okay, so you now join me a whole day later in a sweltering hot kitchen. So I've gone for the lab coat and shorts combo, which is natural for scientists, I believe. Hopefully you've seen in the stop motion video the effect that the golden syrup and the water had on the eggs, if you weren't too distracted by the fly, which seemed to be obsessed with the golden syrup. So now we're going to repeat the process that we did yesterday, and we are going to weigh the eggs and see what effect it has. Now visually you can see an effect. The water egg is much larger and the golden syrup egg has shrunk up, but we're going to put some numbers to that. So I'm going to carefully, I'm going to do the water one first. Okay, so now I've poured away the water and you can see that this egg is a very chunky boy. Because I only want to weigh the egg, I'm actually going to take some kitchen roll and I'm just going to pat the outside dry. This is so that any mass is due to the egg and not due to any water stuck around the outside. But even just feeling it, it feels really swollen, and I'll be honest with you, I'm a bit worried about breaking it. Just going to turn it off. Okay, so scale's on, it's reading 0. Put it on the scale, and that is now reading 83. 82, let's put it around the middle, lovely, 82. Okay, so that means that it has gone up by a whole 4 grams. Okay, so now I'm going to repeat the process with the golden syrup egg. Okay then, that one was a lot more involved. A, golden syrup is sticky, and B, it's really bizarre. I can't quite explain the feeling of having a saggy egg. But you can quite clearly see just how shrunken this has become. Okay, so let's put that on the scale. And it's gone all the way down to 46 grams. Wow. So that's gone down 30 grams. That means that this egg has lost 30 grams of water to the golden syrup. Unbelievable. So that's the egg results. I know that probably this wouldn't have helped you change your mind about how weirdly disgusting yet beautiful this is. Okay, I just thought I'd do an anterior angle because I don't know how much the overhead will show just how much difference there is between these eggs now. This one which was in the water is really swollen, and I'm really scared I'm going to burst it, whereas this egg is just weird. Okay, so we've managed to shrink and swell up an egg using golden syrup, water and the vinegar to take off the shell. We've also seen that with the potato. To explain this, I'm going to go to my explainer, this time in a different direction. So what's been going on? Well, this is all relies with the process called osmosis. Okay, now the definition of osmosis is the movement of water from an area of high concentration to an area of low concentration across a partially permeable membrane. Now notice that I've talked about high concentration of water and low concentration of water. That's going to be important in a second. This phrase, partially permeable membrane, is a bit of a mouthful, so we need to understand what that actually means. So if we break it down, partially means some, yeah, partial. Permeable means let through. Okay, you might talk about permeating or something like that in other subjects. Membrane is just a membrane. For instance, around our cells we have cell membranes, and specifically the egg was one single cell surrounded by a cell membrane. So if I take this example here, okay, I've got a partially permeable membrane down the middle, and remember that partially permeable membrane means some let through. And I've got some sugar or other dissolved substances, it could be salts, it could be other parts of a cell, and these orange dots are the water. So if I think about this, on the left hand side I've got a high sugar concentration because there's more sugar here than there is over here, and a low water concentration. On the right hand side I've got low sugar and high water. Now crucially you can see that these sugar particles are too big to pass through that partially permeable membrane. Okay, they cannot pass through. Only the water can. So we said that osmosis is the movement of water from high water concentration to low water concentration. We can see I've got a high water concentration here on the right and a low on the left, so that means that the water will move overall in that direction. So what's going on with our egg then? Well, if I zoom in on an egg, okay, you can see that this is actually just a cell membrane, okay, which is therefore partially permeable. So if I take the golden syrup case, inside that egg, okay, I've got some dissolved substances, parts of the cell, and outside it's a very sugary solution. Lots of sugar. So if I'm going to put those labels on again, here on the right hand side I've got a high sugar concentration, low water. Here I've got a low sugar concentration, high water. So that means that the water will move out of the egg and into the golden syrup. And that's why the golden syrup egg shrinks down. If I take it the other way round, okay, here, okay, is the water case. Inside the egg, I've now got the higher sugar concentration and the lower water concentration compared to outside. So that means that the water moves in. So, going back to that golden syrup case, okay, this is what happened to the egg afterwards. It shrivels up, okay, and that has the technical term of plasmolysed. With the water, okay, that egg is now swollen, okay. Now, there's a few technical terms here. I originally was thinking about the word turgid, but that really only applies to plant cells, so you could have the word swollen instead, or if it actually bursts, I suppose you could technically say it is cytolysed. So, osmosis, the reason why the eggs shrink and the reason why the eggs swell. Some ideas now to take that experiment on a little bit further. What you could do is you could swap the eggs over. So you could have shrunk the egg with the golden syrup, swollen the egg with the water, and then take the exact same egg, put them in the other ones, and see the process happen in reverse. That shrunken egg swell back up, and that swollen egg shrink back down. Other things you could do is, instead of using the two extremes, water and golden syrup, you could make set solutions of sugary water. Then you might find an egg where the mass doesn't change at all. What you've then actually discovered is the concentration inside the egg. That could be quite interesting. I know this experiment is a little bit more disgusting and a little bit less wow factor than the Skittles one, but I wanted to continue the theme of substances in transport. I promise you that the next ones will be both a little bit more spectacular and a little bit less disgusting. Maybe. I don't know. I might make them disgusting. It's fun. But thank you. Have fun.